Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will cover how to perform exploratory data analysis for compounds datasets. The thing is, what actually is exploratory data analysis? So, the brief explanation is that to identify general pattern in the data. What is the data in our case? In our case, the data is a compound data set, or I would say a list of compounds or a group of compounds. The more, the better. So, what is all about this tutorial? This tutorial is technically the part 13 of the course Computer-Aided Drug Designing, but it is in kind of optional. If you performed it, that's very well. If you skip it, it's also depend on your research data. So without wasting further time, let's start. So we are here. Um, as usual, we will work on the Google Colab, and here is the link how to access this Google Colab. I will drop it in the description section. Once you see this window, you need to come and open CADD project. Then from the list, you need to select part 13, Exploratory Data Analysis, part 13. Click here. Now, once you are here, connect. This is a brief introduction of this notebook and myself. And for some important reads, you can read these papers. Okay. Once the notebook is connected, you need to click these buttons for installation of Kimball client and some import jobs. Remember, I will try to cover quickly because most of the things I have already covered in certain videos. So, if you are watching this for the first time, don't worry. I will tell you which video you need to watch. Okay. Now, the first step is to pick a data set for your protein from Kimball. Now, if you are new here once again, then you may not know how to pick the data. Therefore, I recommend this video, and here in a sufficient detail, I have discussed that how you can search Kimball and what to select, what not to select, how to select. That's it. But here, I assume you knows the Kimball ID of your dataset and I assume that you have watched this video. So we are using some ID from the Kimball dataset. It's here. So the ID is picked, and we have two entries, index 0, index 1, and we use 4315. So this is for the protein family, this is for the single protein. So we will be interested in index 0, this one for a single protein. Also, you can be interested for both. Okay. Most of the time I received some questions that my data is not too much for a single protein. So I would suggest in that case, you need to run this video multiple times for this target, for this target, for this, if there are many for your data, and then make a master file, merge them manually, make a master file and use them in your analysis. Okay, okay. Now you can see we are selecting this index number. So if you are selecting another index number, just change this entry from 0 to that index number. This is the index number. Okay. Now we are going to retrieve this data into this notebook, and let's see. Okay, this is our data, and these are the compounds. Each row contains a single compounds, and total we have 356 rows and 46 columns. Once again, I will not go to discuss each and every detail of these columns because already covered. Now here, if you want to see just the four lines or two lines, so you can see one, two, three, four. So otherwise, you can skip it. Okay. Here we want to see that what is the unique standard type. Standard type is actually a column name in the data. So that is IC50. And what we do here, we save this data into data underscore one dot CSV. So what we saved the whole data without any filtration, whatever. Now, if you want to see the data again, you can see, but I will not. That's okay. Now, if some of the columns contain some missing values, like the IC50 value is missing, so, or if the smiley is missing, once again, if you don't know the smiley, so you need to watch that first video first. So if smiley is missing or the standard type, a value for IC50 is missing for some entry, so we will filter them out. So for that, you need to click to run these cells one by one. Okay. Now we will process this data and we will remove all other columns and we will keep only three columns, the Kimball ID, the smileys, and the standard value. Remember, this is the particular ID of the compound. This is the smileys of that compounds or the structures smileys, and the standard ID means the IC50 values. Now you can see from 256 it is reduced to 245. It means 11 rows were filtered out. Now we are interested to save this. Okay, so we save the data into a CSV. Don't worry, you can click here and your data will be here, and you can easily download it at the end. So don't worry about this. Okay, now we are going to label these compounds into some categories. And here we will use three categories. And if you are interested, you can use four or maybe two or whatever with different names. So my category names will be an active, and intermediate. And my category information is that if the score or the IC50 value is greater than 10,000, call them inactive. If the IC50 value is smaller than 1,000, call them active. And if the IC50 value is in between greater than 1,000 and smaller than 10,000, call it intermediate. That's it. Okay, so you can change these values, you can change this setting, you can change these names and then run it. Okay, so it's done. Now we will see the data which is not reshaped for an extra column. So you can see the IC50 value is 1,000. So it says active. It's more than 1,000, but smaller than 10,000. So we are seeing intermediate, and there are inactive also. And again, the same data, 245 rows and four columns. Now, if you want to save, just click here, save. That's it. Now, now what we are going to do, we are trying to isolate or extract uh, the smileys from from our data set. Why we are extracting these smileys, I will tell you, but this code will take some time, so you need to first I need to click run. Okay, We are extracting this column or the smileys column from the data because we will use these smileys to calculate some properties for all these compounds. Once the properties are calculated, so we will merge the data again. There's not a problem. So, okay, this code may take 3 to 4 minutes. So that's why I'm going to pause the session and will come back shortly. Now it's completed with me and it took almost 2 minutes. So you can clear this log. Okay, now we will import and we will extract the canonical smileys column and then we will, okay, okay. Now what we are going to do here, we are going to calculate the Lipinski descriptor. What are the Lipinski descriptors or what are Lipinski rules? These are four rules, and the value of every rule is a multiple of five. So that's why we also call it rule of five, but there are four rules. So here we will run this cell. Okay, you may see a kind of error. Then what is the error? The error says that unified sample this okay from RD base import RD kit is version. It means the problem is the RD kit, so why not reinstall it? You need to type pipe install RD kit. It should take less than 30 seconds. Okay, 8 seconds. Clear the log. Now rerun this cell. Okay. Now here we are going to calculate the Lipinski descriptors for all the molecules. Just press this button. They are calculated. Now here we want to see the Lipinski data frame. Now if you can see for all the compounds. Now the problem is we have used all the data. I mean the data which also contain the missing values because the missing values was for IC50, so not for the smileys. But the thing is you can solve it if you only want to use those uh, how to say, if you only want to use those filtered data set, then for every step, once it is cleared, we name it something like here we name it DF clean smileys. But when we were generated DF, when we were trying to generate the DF clean smileys, so what we use data 5, sorry, yes, we use the data 5. So what is where we generated the data 5? We generated the data 5 here and its name is data 3 CSV. So you can go back, but that doesn't matter. Uh, okay. Now DF combined, uh, we are going to combine these values with the original file. Okay. Now, if you want to see, come here, click code, and you just say df underscore bind. Let's see. Okay. Now you can see Kimball IDs, canonical smileys, standard values, the class we generated, and there are some NN values because these were actually those NN. So that doesn't matter. The program will not produce any error, and if it does, we will fix it. Okay, these are the molecular weights, log P, hydrogen donor and hydrogen acceptor. Okay. Now we are going to convert the IC50 values to the PIC50 values. What is the PIC50 values? PIC50 value is actually the minus log 10 of the IC50 value. Why we are doing so? Because when we are representing values with the IC50, then the values are showing are highlighting big gaps. Gaps in a sense, you will see one value of 10 in the active, one value of 700 in the active, one value of 500 in the active because they all are below 1000, we call them active already. So in that case, now if you plot this data, so 10, 500, 900, 700, 5, 10. So all these data, the gaps between them is so big. As a result, we call it that the data is a kind of not normalized. So first we are going to convert all the values into the minus log 10, or we are going to multiply the IC50 with the minus log 10, and we will call them P ISO IC50 values. What will happen with the PIC50 value? I have already covered this, so quickly I will cover it again. That the smaller the IC50 is, the smaller the IC50, the better is the value. But once you turn it to the PIC50 value, it becomes opposite. Then the higher the PIC50 value will corresponds to the smaller IC50 value. You will understand how. Okay, just click here and click all these cells. It will convert the IC50 value into the PIC50 value. Now you can see the standard value norm and the values, the reading becomes different. Okay, then we generate our DF final and we name it the PIC50 value. Now you can see 12, 12, 12, 11, 12, and these all are active. And the 11 comes in the intermediate case. So what was the intermediate? Greater than 10,000. Here you can see the difference between the active and inactive is not uh, higher, so it will be easy for us to plot it, and it will be a kind of meaningful. Okay, we have some in n values. I already told you that why, so it's okay, it doesn't matter. Okay, here is our data 4. We save it here, and the data 4 will contain the conversion of PIC50 to conversion to PIC50 value. Now here I'm going to drop this intermediate class now, and I will keep only the active and inactive data. For you, it doesn't mean that you also need to drop the intermediate class. You can drop the active, you can drop the inactive. It's up to you what two classes you are going to combine. So you can try by yourself. And okay, I'm going to drop the intermediate class, and then, oh sorry, okay, you can save this to data 5 where we will not see any intermediate class. Then now we are going to perform some exploratory data analysis or chemical space analysis, and we will use the Lipinski descriptor we calculated for every compound in these analysis. Now, the first thing we are going to plot or we are going to analyze is the frequency in the data set of the two classes. First class was active and the second was inactive because we have removed the intermediate class. Now, if you come back here and you remove the active, and let's say I'm going to remove the active for you. Let's say remove the inactive just to show you. Click here, remove the inactive, then rerun all these things because here I skipped this. I don't want to save it again. Now, as we have removed the inactive, then you can see active and intermediate. So this portion was removed here. Now again, I'm going to come this to come back to the same pattern. Okay, you can see now inactive. Okay, now if you want to save this data in a PDF, just click here and you will have this figure here. You can see here, you click here, download, and let me show you that. Oh, okay, probably some problem with the PDF converter. That's not an issue. I will fix it. Okay. Now, the important thing is how to perform these analysis. These are smaller issue. I will fix it and update uh, the notebook. Okay. Now we are going to perform some analysis between those descriptor. This was the dataset analysis between the two classes. Now we are going to perform analysis between different groups, and we have four groups. Okay. So first we click here and we see what analysis we are seeing. We are seeing two different colors. One is the blue for active, the orange is for inactive, and we are seeing the distribution of PIC 50 value. The PIC 50 value is based on the size. So if a smaller size, let's say this one compared to this one, or compared to some, okay, this one and this one. If you see, so the smaller size means the smaller PIC 50 value. The bigger size means the higher PIC 50 value. And I told you the higher PIC 50 value means the compound is more towards the active region. Okay. So, and on the x-axis and y-axis, we have plotted that the molecular weight here on the axis, and on the y-axis we have the, how to say, the log p, or I would say, okay. Now if you want to change the some things, the fonts, the size, you can tweak this option. The figures, each and everything are mentioned here. Now, this graph, you don't need to go quickly down because you can regenerate this graph for different settings. Come here and just see the data. We have log p, we have molecular weight, we have number of hydrogen donors and we have number of hydrogen acceptors. Now, let's say I'm interested to generate a graph between the molecular weight and the number of hydrogen donors. How can I do it? Or how can I generate this graph? So here you can say molecular weight, keep it the same, and num h donor. Okay, one thing you need to take care of, please keep the same spelling and italicization, whatever, NUMH donors. Because if we run it like this, it will generate an error. You see? Now, donors. Okay. The graph is okay. You will be surprised that it still say log P, but I changed the donors here, but I did not change it here. That okay. Now you can see it's the number of hydrogen donors and the number of molecular weight and the PIC 50 is still the same. So you can regenerate these plots and you can let me check whether it's same or it's okay. Here we are able to generate the plot and we are able to save it. Okay, if you are not seeing this legend, doesn't matter. You can tweak the figure options or the figure size. It will be fixed because I have selected 5.5 and 5.5. You can increase it or you can decrease it. Then this way you can fix these issues. Okay. Also, you can play with edge color, you can play with other things like the hue class. Okay, in some other video, I will try to explain these things in matplot, but I'm afraid it will not be an interest for you. So, therefore, simply I would suggest to play with the figure size. Okay, now we are doing some calculation. So, let me remind you that you can generate almost a variety of plots here. Um Molecular weight with number of hydrogen donor, molecular weight with acceptor, molecular weight with log P. 3, then log P with these two 5. So almost you can generate 5 kinds of analysis here and then say one by one. Okay, now we are going to do some calculation with box plot. This was called the scattered plot. Now, what kind of calculation? This is an important calculation. If you are performing drug designing on this data set, then this figure will help you. For example, you are predicting PIC 50 value for some unknown compounds. Now, maybe you are asking me how to predict that PIC 50 values for unknown compounds. So I have already covered a tutorial number video number 11 where we have used artificial neural networks for the prediction of PIC 50 value. So let's say you have this exploratory data analysis on your data set. Now your compound generates an unknown compound, I would say, or a new compound from the PubChem or anywhere else. You used it and it generates the PIC 50 value of, let's say, 11 here. Now you compare that PIC 50 value with this figure. So I would suggest that don't waste much time with that compound which generated PIC 5011 because it falls in the inactive region. Can you see here? But if it is 13 or more than 13, it means it is a potential hit. It is a potential compound. So that's why I would suggest, if you are following those videos, so also do these exploratory, or also perform these exploratory data analysis. It will help you to identify compounds based on the um, PIC50 value, that where it lies. Okay. Once again, I will drop a link of that video here, so it will not be a problem for you to search it on the channel. Okay. Now you will see here the link. Okay. Now here we are going to perform some statistical analysis. We call it Mann-Whitney U-test. And what is the Mann-Whitney U-test? You can read for yourself, but let me give you a simple description. Mann-Whitney U-test actually tests the null distribution or the null hypothesis. So if it answer you that the null hypothesis is true or rejected, it means the result is positive. But if it says that the null hypothesis failed to calculate or such kind of things, then it means there is no significant difference in the comparison. Okay, so now we are going to compare the distribution of these features. I would say not feature, because in machine learning we call it features, but the distribution of these properties, these four properties, that whether they are significant or not. If they are not significant, so I would not recommend to just base your story on those, on these features. Or if some of the feature are insignificant, so you try to not focus on that specific feature. Okay, so let's perform a statistical test, and then click here. Here we perform a statistical test on PIC50 values, that the PIC50 values between the two classes, is it significant or not? So yes, it, you can see reject. So it means it's significant. Now we are going the molecular weight analysis, and uh, then the active class. This is the molecular weights. These are the molecular weights, and then this is the inactive class. So let's see. Yes, you can compare it, you can rely on the molecular weight. Now log P, or solubility. So for the inactive class, it's here. For active class, it's here. So let's see whether, yes, it also satisfy the Mann-Whitney U-test. Now for the donors or acceptors, so active and inactive. Uh, let's see, fail to reject. It means that between these two classes, active and inactive, do not rely mainly on number of hydrogen donors or acceptor, because uh, it may not give you a clear picture. Because if you also visually you can see, um, it makes no difference. Status also visually, so it means statistically is for sure it makes no difference. Okay, so you can also see C fails to reject the null distribution or the null hypothesis, and this is for the number of hydrogen acceptor. And let's check. Okay, so if you are using this data set, let's say, then do not rely on this number of hydrogen acceptor and number of hydrogen donor further, and you can rely on the relation on the values of the PIC50, log P, and molecular weight. Okay, now here is the some interpretation, and you can read it. I have already discuss. Now I want to download all the analysis in zip. So you can click here. You will see it will be downloaded here. Wait. Okay. No, sorry. Exploratory data analysis.zip. It will be generated here. Here. Now click download. One thing you need to know that is this one, this one. Yes, because you can generate five or six plots, or you can generate five or six different analysis from this section. So for every analysis, you need to change the name of the figure here. So it will be saved here, and then when you change or you perform another analysis, then change it again. So it will not be overwritten, and at the end, when we click the zip stuffs here, then all these files will be included. So let's see what we have downloaded. Um, Exploratory data analysis. I will cut it from the download section and will bring it to my desktop, and let's see, paste. Okay, make a new folder. Let's say okay, drop it here. Now I will just extract. Okay, exploratory data analysis. So you can see the data 1, 2, 3, 4, 5. We saved it, and those man Whitney test we perform, and some plots we performed. Let's say this plot. Let's say this plot. Okay. So I think it was interesting, and it will be uh, of some use in your research. If you are liking my work, if you want to continue this work, if you want me to continue this work, you can subscribe this channel. I would suggest to subscribe, as I already mentioned before that almost 20% are subscriber in the watch time, and 80% are not subscribers. So if you are watching, why not you are subscribing? Okay, you can also follow my WhatsApp channel, and you need to just click here. Once you click here, you will see this message. You click here, and then you will see this channel, Benfolab. You can view this channel, and then you can, how to say, you can follow this channel. Okay, I'm not going to show you further. Okay, so you can follow this channel. Why, what are the benefits for you in this channel? Sometime I will share some updates here, so it will reach you in time. Another thing, I also offer some research services, so you will also see that in case you or your fellow is interested. And sometime I also offer some courses, so it will reach you. Those courses, the information will reach you for free. So, and also, I don't know, because I'm new to the WhatsApp channel, that whether you will be able to message there or not. So I can't say that you will talk to me there or not, but at least you can see my updates. You can see my research services and courses. Okay, thank you so much, and thank you for your time. Take care, and good luck in your research. Bye bye.